Hello.
How are you, Anyara?
I'm good. How are you?
I'm good. Thank you. What's new?
Not much.
Hmm.
This week I've really started doing the 30 questions a day in UWorld that we like sort of agreed on last time.
Yep.
Um, I only focus on bio. So I was just doing bio every single day.
Mhm.
Uh, and I think it helped. Like I could see that I could start picking up like what I sort of needed to know from the passages just by the repetitiveness of it.
Yep.
So I think it was good. I don't know if you would recommend only focusing on like one subject throughout the whole week, which
Hmm.
I did. But I think that like just being surrounded by bio helped.
Yeah, yeah. Because I know we talked about the sciences being the primary thing to to work on, and we talked about content gaps in bio regarding like physiology and stuff. Um, so yeah, it's good that you're getting through that. I think it's okay that you're doing just bio for now. Uh, and yeah, based on what you told me. So that's good. Yeah, how's that going?
It was tough, not going to lie.
Yeah.
Just um a lot of those questions were like content that I have to review, so
Yep.
a lot of content.
Yeah, totally. All right, what would you like to do today?
I did find like some like I wrote down some specific questions that I wanted to review.
Mhm.
And just if I had to choose like one topic that I got most of the questions wrong in, were that would be genetics. So
Yeah.
if we could like go through some genetics or like what I need to know for genetics, that would be great.
Okay. Yeah, let's do that. Yeah, genetics can be pretty tough. And when you say genetics, like, there's a lot of things that could be tested. But yeah, you can show me the questions.
Funny enough, I don't have any genetic questions here, and it's because I
Oh.
understood the like explanations, so
Mhm.
I didn't write it like I didn't get them. But how should we do this? So can I maybe I can just I screenshotted the questions. Um, or yeah, maybe on the chat.
You can share your screen if you want.
Sure, let me try that too. Um, Video settings. No, full screen. Share screen. Thanks. Okay, can you see my notes?
Mhm.
Yeah.
Got it.
So,
Yep.
um, yeah, so that's like sort of the first question I had was this question on so let me know if it's better if I open up the passage because I can. I have the test IDs here, but I just screenshotted the questions themselves.
Got it.
Mhm.
Um, yeah, so this seems to be talking about um well, I guess the muscle. It's just that the CAM complex and the MLCK are not like discrete things, so that stuff would be related to the passage. Um, so yeah, you can open up the passage.
Yeah, so let me just go.
Okay.
I don't mind just sharing my screen in general, since I might be going back and forth.
Mhm.
Let me how I can do that. Here, share screen. Um, Advance. No, I guess I have to choose what to share. Okay. Can you see this?
Mhm.
Okay. So I think this is 33127. Find that. Never really looked for. Do you know how to find the test ID?
Oh, I thought you had um oh, you're saying to search for it by test ID?
Yeah. So
Uh,
I like wrote down here the test ID. Now I'm,
uh-huh.
I
What
guess
happens if you put it into that?
here.
Yeah.
No,
Whoops. Okay, let's see. Um,
it's probably going to be this one. Let me just see. Copy test ID and one. Is it the same one?
oh yeah, yeah, and then you could just paste it anywhere just to see if it's the same one. Yeah.
Nope. It's not that one. Maybe this one. Yep.
That one?
I can click.
Oh, you'll hit review.
Oh yeah.
Mhm.
That's fine. I think it was 11. No. Yeah.
Okay, here we go. So, okay. Aside from just the content, I want you to read the passage out loud, and I want you to tell me, like, think out loud also. Like, tell me what you're thinking of as you read this.
Okay. So I'll start reading it.
Mhm.
So potentiation is a phenomenon in which skeletal muscle force production in response to the same depolarizing stimulus increases during a series of contractions. So you have the same stimulus, and you're increasing your force production. So it's kind of reminds me of like becoming more sensitive, like sensitizing. Uh, the activity of an enzyme called myosin light chain kinase is thought to play an important role in potentiation. So I think of enzymes or probably some speeding up a reaction or something like that.
So, there's a something that we call buzzwords, which are words that you see in any given MCAT passage that they'll likely ask you questions on. So for this passage so far, I want to ask you about kinases.
Yep.
What can you tell me about kinases?
So kinases phosphorylate. So
Okay,
they're really involved in a lot of the times like activating or inactivating. So they can either like phosphorylate or dephosphorylate.
okay. What enzyme class do they fall under?
Um, what do you mean by enzyme class?
So for the enzymes, you want to know the classes. So like for instance, um, there are oxidoreductases, which are enzymes that cause redox reactions. Yeah,
Oh, I see. So would it be a transferase?
exactly. Now, tell me so like, what does that mean, a transferase?
So you're getting phosphate group probably from ATP. Um and you're placing it in another molecule.
Huh?
Yep.
Yeah. So you're just transferring a phosphate, right? Now, okay. Now, kinases require like three specific amino acids to work through.
Hmm.
Do you know what I'm talking about?
Vaguely remember something like this, but no. Uh, is lysine one?
So it's going to be sty.
Serine, threonine, tyrosine.
Yeah.
So because of the OH groups?
Mm-hmm. Because yeah, they have an OH group. Now, what what can an OH group act as?
Um, well, it could remove a proton.
So, uh, I think you're thinking of maybe closer to like an OH minus, like a hydroxide. But if the OH is still attached to something, right? So, okay, so this is going to be the most common nucleophile that you'll see on the test. What do I mean when I say nucleophile?
Do you mean that it can act as a nucleophile like in a reaction? Like it can like attack and I guess an electrophilic center, but donate electrons?
Yeah. So a nucleophile is so it's kind of like in contrast to what you said earlier about um deprotonating something like we can think of so when generally speaking when we talk about acids and bases we are talking about the Bronsted-Lowry definition. Like an
Mhm.
acid is defined as the as something that donates protons, and a base is defined as something that gets protonated. But when it comes to organic chemistry, we're concerned about Lewis bases and Lewis acids. And so a nucleophile, would that be a Lewis acid or a Lewis base?
I feel like
Or,
it's a
or
big
you could tell me like so what is a Lewis acid and what's a Lewis base?
um so does a Lewis acid accept a proton?
Well,
A Lewis base donates a proton?
well, it can't be, you know, it can't be like contradicting the Bronsted-Lowry one definition, right? Because like if something donates protons, like it's going to be an acid. Um,
Okay.
so uh so we can view Lewis acids and bases as, you know, being something like, a little different, but it won't, contradict the Bronsted-Lowry definition.
Okay. I guess I was mixing them up, like
Mhm.
this definition and that definition. So if you accept a proton, are you donating an electron?
So if we have, let's think of it like this, right? Um, so the word nucleophile, right? What does that mean?
Um, I think it means nucleus loving.
Yep. Now, what can you tell me about a nucleus?
It's full of protons,
Uh-huh.
it's
So it has a what charge?
positively charged.
Okay. So a nucleophile will like that positively charged thing, right?
Yes.
So what can a nucleophile be? Like what can it do to the positive thing?
It can stabilize it.
Yep, how can it do that?
You sorry, you said the positive thing, like the
Oh, sorry, yeah. Um so what can a Lewis base do to stabilize like a positive charge?
um. So the Lewis base is a nucleophile from what you're telling me, and um, if it's nucleus loving, which the nucleus is in itself positively charged, um, I feel like it could, if it's positively charged, wouldn't it be attracted to a negatively charged thing, um, to like neutralize like positive and negative?
Yeah.
Uh, yeah.
So a Lewis base will be a donor of something. What do you think it'll be a donor of? Like what can you donate to something that's positive?
Electrons?
Yep.
Okay.
So a Lewis base is an electron pair donor,
Okay.
and a Lewis acid is an electron pair acceptor, right? And and the Lewis base is also the nucleophile,
Okay.
and the Lewis I think I don't know if did I share like a Google Drive folder with you?
No.
Oh crap. Okay, I'm gonna share that with you like, ASAP because I have lots of stuff in it um that can be useful. And one of the I have like a PDF that I just kind of explain Orgo in like as simplest terms as possible, but Yeah, organic chemistry is like, if I were to summarize it, it's the interaction between nucleophiles and electrophiles, which means it's the interaction between Lewis bases and Lewis acids. So the most common nucleophile you will see on the test will be a hydroxyl group,
Okay.
because it would have two lone pairs, right?
Yes,
Now, the most common electrophile that you will see on the test will be a carbonyl group. Let's
I
say
see.
this is, yeah. So carbonyl is going to be what?
And electrophile.
Oh yeah. But like specifically, like what is a carbonyl group?
Um, like like in terms of acids and bases.
Oh, sorry, just like the structure, I guess.
Oh, so you have like carbon with a double bond to oxygen.
Good, good, good.
And so
So,
you have,
yep.
oh, so you
Oh,
have like electron density pulled towards the oxygen, and
exactly.
the carbon is an electrophilic center. Mhm.
Yep. So we can say that it would have a partial positive
Yep.
charge, right? So
Yes.
like everything that you know about charges, so this is like, you know how when they tell you to like memorize the amino acids and they tell you like these are the polar, these are the non-polar.
Yeah.
Um, I think it's the best way to think about it is um, first of all, which amino acids are fully charged, and that'll be like the acidic ones and the basic ones. And then after that, instead of thinking of polar or non-polar, understand that polar simply means not a full charge, but a partial charge.
I see.
So, so that means that you can remember the five amino acids that are charged, and then the five amino acids that have partial charges, which make them polar, and then the rest of the 10 must be nonpolar, right? So, yes, so that means that there's a partial positive at this carbonyl carbon, a partial negative here. I mean, the overall dipole is like that.
I see. Mhm.
So, okay, so what will this hydroxide do here?
So I can nucleophilically attack the carbonyl carbon
Yep, so it would go there, right? And this would go up here. Why would that go up there?
because of resonance, is it? Or
Well, um, maybe there's another reason, uh, more straightforward one.
so you're like attacking and breaking up that double bond. So you have a tetrahedral intermediate.
Yeah, yeah, yeah. Because carbon can only have four bonds, right?
Yes.
And here carbon's already bound to four different things. So if the hydroxide binds to it, it'll be like five different things. So you want to get rid of. So like this would look like um something like this. Like, okay, the OH here, that, this O minus, and then this leaving group here. Um, and it's going to want to go the electrons want to go back to create this double bond, and it'll kick off this leaving group, and that would give you, oops, something like this.
I see. Yeah.
Okay, good. So just like the carbonyl looks like is a good electrophile, um phosphate, oops, yeah, phosphate looks kind of similar here, right? You have like a phosphorus that's double bound to an oxygen, and it has even more oxygens around it, pulling electron density, right?
Yeah.
So the kinase can do the same thing here, right? and then this comes back down, and then this breaks off.
Yes. Wait,
So
let
yeah,
me try to process it.
sure.
So yeah, beacon attack. I see. I see. Okay.
Yep. So 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 since a kinase is a transferase that transfers phosphate groups, it has to use either serine, threonine, or tyrosine, those amino acids, to facilitate the transfer of phosphates.
I see. Um,
Hmm?
I see. Kinases. Okay. I think I'm vaguely remember, like, are these also called like phosphomimetic?
So
Acids?
good. Yeah, it's good that you asked that. So, um, well, uh, so phosphomimetic would be like okay. So let when this hydroxyl gets phosphorylated, does its charge change?
When it gets phosphorylated.
Hmm. Or so phosphate, does that have a charge?
Um, it's usually negatively charged, right?
Huh? So if something gets phosphorylated, it takes on a negative charge, right?
Yes.
Now, a phosphomimetic thing, it does not need to be phosphorylated, but it can act as if it's phosphorylated, which simply means that it would also have a negative charge. So if they were to ask you like what could be a phosphomimetic, amino acid, it would be like the negatively charged ones, like aspartic acid or glutamic acid, right? So phosphomimetic is going to be something that will functionally behave like it's phosphorylated in that it will have a negative charge. Yeah.
Okay.
Hmm.
I see. So,
Yep. Of
okay. Sorry, I'm just trying to absorb everything.
course. Yeah.
Yeah. Okay, sounds good.
Yep. So, um, okay, yeah. So basically, nucleophiles are Lewis bases, which mean they donate lone pairs. Most common Lewis base, most common nucleophile you'll see is a hydroxyl group. So always be, on the lookout for that if you see, hydroxyl groups and stuff. I mean, even, how, so what's more stable, DNA or RNA?
DNA.
Yep. Why do you think?
Um, um, I think there's something with a methyl group.
So, uh, so what's the difference between DNA and RNA?
It's an OH group. Versus and is an OH group versus a methyl group? Is that correct?
So, so, so which one would have? So, okay, let's start with DNA. So that's deoxy, right?
Yes.
So RNA should have a not like more hydroxyl groups than DNA, right? And we know for DNA, like one, two, three, there's a three, there's a hydroxyl at the three position, right?
Okay.
But
I have to review the structure, but yes.
oh yeah. So you know, like you don't have to know this. I mean, sure you do, but it's going to be some type of like ribose sugar that's bound to some like purine, pyrimidine base, and then phosphate.
Yeah.
So okay, now yeah, the ribose sugar will have a hydroxyl group on the 3' end. That's why, like, DNA has a 3' end, right? Because when DNA gets polymerizes, the 3' hydroxyl group attacks the phosphate group of a different nucleotide, just like it does here. That's how it can make the DNA, right? So, okay, so this is going to be like DNA, whereas RNA will have an OH group at the second position also. Now, these are really, really nucleophilic things right next to each other. So what can happen is this can attack that and cause, the RNA molecule itself to react with itself, messing it up and stuff like that. So that's why, that's one of the reasons why DNA is more stable. But um, but yeah, so kinase buzzword. Anytime you see it, you want to think STY. You want to understand that it's a transferase. Um, and you know, and what that means is that it's defined simply by transferring phosphates. It doesn't mean that it will always phosphorylate something. It doesn't mean, like, for example, glucose can get phosphorylated into glucose 6 phosphate, right?
Mhm.
So that'll be like you're taking it from ATP to, produce ADP.
Yes.
But then when we have PEP, pyruvate, enol, um, phosphate, uh, turning that into pyruvate, it also uses, so like, okay, like this is hexokinase, right? But this is going to be called pyruvate kinase, except this time it's actually removing a phosphate from PEP, right? So this time, actually, you're going to have an ADP turn into an ATP.
I
But
see.
this just shows that um, that a kinase isn't always going to phosphorylate your substrate. It can dephosphorylate your substrate and give it back to ADP to make ATP.
I see.
Yep.
I-
So yeah, so good. Um, any questions about this?
I'll try to recap.
Yeah.
Uh, so what you're telling me is that kinases usually phosphorylate these specific amino acids as STY in a protein.
So sorry. So the kinase itself, it's a enzyme, so it's a protein. It itself at its active site will have ST and or Y, right? And that will enable it to take a phosphate group from something and momentarily give it to the kinase. So it can be from ATP, it can be from some substrate, but the STY is a part of the kinase itself.
Oh, I see.
And then it can give the phosphate to, a molecule.
Oh, I see.
Mhm.
Okay.
Yep. And yeah, you'll see tons and tons of questions about kinases. So it's a very good, high yield kind of thing.
Yeah.
All right. Any other questions so far?
Nope. Good.
Okay. So um if you want, you can screenshot this, but I can also just, you can take it from like the recording.
Okay.
Um,
Yeah.
yeah.
I think I'll take it from the recording. Yeah.
Okay. So all right, so I'm gonna clear it. All right. So cool. So we have a kinase that we talked about. All right, you can continue.
So MLCK phosphorylates a protein called the myosin regulatory light chain,
Okay.
which is part of the myosin molecule composing the thick filament.
Mm-hmm. Yeah.
The activity of
Oh,
this,
sorry. So I was gonna say like, yeah, so tell me like what you're thinking.
um, I got really confused when they mentioned the thick filament. Uh,
Okay. But that first part, MLCK phosphorylate something, right?
yeah,
Makes sense because we are dealing with kinases, right?
yes.
All right. And some type of light chain thing. Um, but okay, let's talk a little bit about thick and thin filaments.
Oh shit.
Mhm.
Uh,
Oh, crap. Oops.
muscles, but I don't really remember any of that.
Yep.
Uh,
Okay.
mhm,
Yeah. So basically, you're going to have like myosin, and you'll also have actin.
yes.
Now, um, so myosin is going to be part of what we call the thick filament, and the actin is going to be what we call the thin filament. Now, um, when it comes to, I guess like, so this is like the sarcomere kind of thing.
Mhm.
So essentially what that means is, well, I guess we can continue with the passage and then, because I'm gonna go have to likely go over it anyway for this question, but the thick filament, think of so it's made of the myosin, and yeah, the thin filament is made of, actin.
Okay.
Yeah.
So the activity of this myosin light chain kinase is regulated by the binding of the protein calmodulin. Okay, so calmodulin is a modulator of this kinase. So it's
Hmm.
modulating an enzyme,
Yep.
which occurs when calmodulin binds calcium released from the sarcoplasmic reticulum.
Hmm.
So I know that this is sort of like, or I think of like muscle contraction initiation when I think of like calcium being released from the sarcoplasmic reticulum.
Yeah. Go on. Tell me more about that.
Um, yes, so calcium is sort of stored in this structure
Hmm.
and
Which structure?
in the sarcoplasm here
Uh-huh.
in the sarcoplasmic reticulum.
What is
And
that?
um I feel like it's like something that surrounds the sarcomere, like
Yep.
and is like a bunch of
Hmm.
I'm thinking T tubules, but I don't know why that's coming up. Like it's like
Well, that's part of it. Yeah.
part of it, yes.
Yep.
Um, and it's sort of it's full of calcium and it can reuptake it really quickly afterwards as well.
Hmm. Yeah. So you can think of it essentially as like the endoplasmic reticulum within, or that I guess surround the muscle, the muscle, the sarcomere. Okay. Now, and yes, it's going to be like connected to like a T tubule, um, which is what?
Uh, I don't know. I remember the word, but I don't know what it is.
Oh yeah, yeah. So I'm not necessarily asking like, to give me, you know, an answer. I just want you to think out loud, essentially, to tell me what you're thinking. That will give me a good idea of like, your thought process and stuff.
Yeah.
So yeah, what can you, yeah, tell me what you think about when you're telling me about um the T tubule and sarcoplasmic reticulum and the calcium and stuff like that.
I just think of uh muscle contraction and calcium stored here and released from this uh maybe through the T2s. I don't know. I just don't remember.
Whoops. Okay. Yeah, yeah. So basically, uh, sorry. Uh, I was gonna use, okay, whatever. I was gonna see if I should get my tablet out to draw something, but basically, yeah, let's say that, this is some sarcoplasmic reticulum, and this is like the outside of the um muscle, and that's like that, and that's like that.
Is it involved in something like connection, like T-tubules connecting? I think I'm getting some of that from my distant memory.
Hmm. Yeah, so like this would be like these would be like the sarcoplasmic reticulum, um and yeah, this is the T tubule. You could see that, it's forward as a T, and um whatever, there'll be like these transporters here, so calcium can go, out of the sarcoplasmic reticulum into the T tubule and then, outwards. And this is like at the neuromuscular junction, right? So like, this is like all muscle here, like muscle, muscle. But then you can have your, you know, your synapse here, so what happens is that calcium ion gets taken up by the the synapse here. And what that causes is for the neuron here to release a neurotransmitter like acetylcholine. And then that can, exit and be in this neuromuscular junction. This the muscular part is like the motor end plate. That's like one of the names for it. And um and yeah, the acetylcholine can bind to ligand bound, to ligand gated ion channels. And then it can cause like, an influx of the sodium to have that depolarization, action potential continue on.
Yeah.
Um, so that's yeah, so that's kind of like a simplification of it. Um, but calcium also plays a role in because like okay, like this is just at the neuromuscular junction, but then at the level of the muscle itself, where you have, your thick filament, your thin filament, um, what happens is that the um the thin filament, that's composed of actin, like kind of looks like these beads that are kind of in like a helix. The actin filaments, they have something called tropomyosin, which kind of the covers the sites on the actin that um myosin can bind to. So, but what calcium does in this is that it rearranges the tropomyosin to expose the myosin binding sites to let uh myosin bind to it, and then, start that um muscle contraction.
Okay.
But um but good. So yeah, calcium released from the sarcoplasmic reticulum. You understand that. Yeah, and you can continue.
Mhm. Okay. Binding of the calcium calmodulin complex triggers rearrangement, which is of MLCK, in which an autoinhibitory region moves away from the active site for RLC phosphorylation. That's a lot
Okay.
of. Me and packet. Um, So MLCK is normally inhibited, but it can be uninhibited. It can be activated by this complex binding, the
Mhm.
cosmodulin binding.
Okay. So when the calmodulin, binds to calcium, it causes a rearrangement of the kinase. And so there's some type of auto-inhibitory region of the kinase that when, the calcium calmodulin complex binds to it, causes that inhibitory region to move away from the active site for RLC phosphorylation. And that's the, you know, myosin regulatory light chain. Um, so okay, continue.
RLC phosphorylation, which has been shown to vary with the frequency and strength of contractions, results in repositioning of the myosin head slightly closer to actin binding sites, thereby facilitating actin-myosin interactions. Slightly closer to the actin binding sites. Which is, yeah, so you mentioned like myosin closer to actin is going to facilitate contraction, like so phosphorylation is helping them be closer.
Yeah, so this is, in other words, what we talked about regarding the tropomyosin and stuff. Um, it's just using different language to make it seem like it's new. Because like the MCAT, they want you to feel like, oh, I didn't study enough, you know? Like when you see myosin regulatory light chain, what they want you to feel is, oh, I don't remember that, I didn't study that. But that's, you're gonna see that type of thing. And the answer to it isn't that you need to know it. The answer to it is, does this fit in with what I already know? Right? So if we're talking about you know, some type of calcium attaching to something um which makes it which causes an end result of the myosin being closer to the actin binding sites, then that's pretty much, aligned with what we know already. Just they're just using more confusing words,
Yeah.
right? Um okay. So so I guess so like what we can take away from the second passage is calcium gets released and it leads to enhanced actin myosin interactions.
Yes.
So yeah, maybe I would like um just like highlight that and that.
Okay. Yep. Okay. I guess should I continue?
Mhm.
To investigate the role of myosin RLC phosphorylation in skeletal muscle function, researchers studied in vitro force production by the EDL, a fast-twitch skeletal muscle of wild-type mice and mice with no skeletal muscle. Okay.
Okay. So now, this is again typical of these passages. Now they're talking about, an experiment. Um, and again, they have an excess of, or it's a very verbose, way of writing it, of writing this. But again, think of that as just, the test is designed to psych you out. So don't get too worried about or bogged down by like, the specific muscle of EDL and stuff like that. What we really want to see is um that if we want to see like how the phosphorylation of myosin plays a role in, the in muscle, they compared wild type mice and then mice with no MLK. So that's the knockout.
Okay.
Okay. All right.
The EDL muscles were electrically stimulated to produce several very, very brief twitch, submaximal contractions, each separated by a minute of rest.
Okay. Now, why do you think they are separated by a minute of rest?
Um, so you could have potentiation probably. Um, if you simulate them very close together, you
Mhm.
could have like an enhanced muscle contraction.
Okay. Um, so 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 okay, when we have something like that, you can have um or let's think about, I guess, in just neurons, not necessarily muscles. Um, you know, let's say that you're stimulating, neurons and um but you would need to separate it by a minute of rest. I guess what principle is that? Or uh I
Yeah.
guess yeah.
So maybe you're referring to like you have a refractory period.
Yes. Yes.
Um, mhm.
Yep. Exactly. Yeah. You have you have a refractory period. So that's why, they're going to separate it by a minute of rest. Um, okay, cool. Yep, you can, yeah, continue.
The EDL muscles were, yep, sorry, I already read that.
Mm-hmm.
These baseline contractions were followed by a much stronger sensitizing contraction, followed several seconds later by another twitch contraction.
Okay.
Okay. So the first ones were baseline, and now they're giving them stronger contractions.
Yep, yep. And yeah, we can see that when they mentioned the Twitch ones, they said submaximal. Uh,
Yeah.
so that just indicates that it's not strong, and so then it makes sense that then they did, the stronger one.
Mhm.
All right, good. Continue.
Within each set of mice, the forces of the baseline twitch contractions were similar. However, the final twitch contraction was significantly elevated in wild type, but not the knockout mice muscles.
All right, yeah. So that part's big.
Yeah. So
So
my yeah,
yeah.
I can just go over what I think. So the first paragraph they were mentioning how this was involved in the potent they mentioned potentiation. And so when you don't have it, you won't have that increased contraction. You won't have that elevation. So it's sort of proving what they thought was the case.
Mm-hmm. Yeah. So we talked about well, we talked about so something. Okay, so let me show you like kind of like a strategy that is like I call it like drawing like a schematic. So like for instance, let's see how I can do this. MLK causes um the RLC to become phosphorylated, and they mentioned the calcium and And then I'm going to also do like a proportionality type of relationship. So I'm gonna say that, you know, calcium 2 plus whatever cam complex is going to be proportional to actin myosin interactions. And then then we have, oh yeah, then I could have really just done that because the RLC phosphorylation. So yeah, essentially, so from here we see that MLK phosphorylates RLC, so I drew the phosphorylated RLC, and CAM, you know, can facilitate these interactions, so I have that here.
Mhm.
And then also RLC phosphorylation also facilitates the um interactions, right?
Yeah.
And then they mention about, okay, then they're comparing the muscles for um regular mice and muscles for the ones that don't have the MLCK. So if they compared the two, they found that, so maybe I'll say like Twitch, whoops, Twitch contraction is going to be um larger, or yeah, Twitch contraction, like I guess final Twitch contraction is larger, um so I'll say proportional to wild type. um or I could have just said that the mutant has an inverse proportion with that final Twitch uh contractions. So I don't know if I talked to you before about like proportionality stuff. Oh,
No.
yeah, there. Okay, so yeah, so basically, so let's say A is proportional to B, that means as A increases, B increases. If A decreases, B decreases. So that's going to look like that. If
Yeah.
it's proportional to the square, it will look like that. And now the inverse proportion is one that they like to ask about, because what do you think that would look like?
Um, the opposite of the first, so decreasing, like when one increases, the other decreases.
Like that?
Yes.
So let's try some numbers, right? So if A is 1, B would be 1. If A is 2, B would be what?
Uh, B would be if A is 1, B would be 1.
Oh yeah, sorry, I mean like the 1 over B part. Yeah.
Okay, so if A is 2, then you would have a half.
Okay. And if this is 3, this would be 1/3.
Yes.
And let's see, something down the line. So what if we had 300 for A?
It would be a very, very small number.
It would be 1 over 300, right?
Yeah.
So so as A increases, B decreases, but what number can this be not be?
0.
Yeah. So what happens here is, do you remember what asymptotes were?
Yeah, so it's approaching it, but it never reaches it. So I guess would that make it like sort of like inverse of the exponential one?
Kind of.
Um,
Kind of. So we're gonna have asymptotes at the axes, and we're going to have therefore something like this. So it's essentially the same line, except as you approach the axes, that's when you're going to have that curve, because it's going to infinitely approach the axes, but it'll never touch.
I see. Yep.
So it's kind of like if you, like, let's say you walk to that door or whatever behind you, and each step you take is one half of the previous step. Will you ever reach that wall or your door?
No.
Correct. So, yeah, so thinking in terms of proportionalities is a super good strategy for a lot of things, especially this test. And also being familiar with how things would look like if they're graphed, because they like to ask. This is a favorite question of theirs because they want you to choose something that just looks like that.
Okay.
Mhm.
I felt on the trap.
Yep, yep. Yeah, we all did. I remember when I did as well. But okay, so what I'm doing here with the proportionality symbol is that, the more calcium, calmodulin type of, you know, interaction here, as that goes up, so does actin myosin interactions. And then here I wrote for the mutant knockout, there's a smaller final twitch contraction,
See.
right? Because
Mhm.
I could have also just said, you know, final twitch contraction proportional to wild type. Um, so this way I just have it in both ways. So the mutant, um, will have that inverse proportion. Okay, cool. All right, you can continue.
Um,
Oh,
uh,
you're here.
so yeah, the average force produced by the twitch contractions before and after the sensitizing contraction are shown in table 1 and are expressed relative to the average baseline force in wild type muscles. So they're normalized. I
Yep.
see. Um,
So, so whenever we're, interpreting data, I would always say start off reading with reading the caption.
okay. Like this table 1, force produced during twitch contractions before and after the sensitizing contraction.
Uh-huh. So what's sensitizing contraction?
So it's the larger dose one that they gave.
Mhm.
Uh, uh, yeah,
Good,
here.
good, good. So, all right. And then when we look at something like this, we just want to go like, don't don't like rush through it. Like, don't, I think it's best to just do it in a slow, systematic way. So I would just start off by, or yeah, tell me how you would interpret this.
So so conditions, so they have the wild type and the knockout ones, before and after, before and after. Before is the submaximal, so it's the baseline one that they're normalizing, which is kind of all, I guess because they're normalizing to this. So that's why it's 1. So it's 1/1, and this is 0.9 over. So it's whatever it was over this. So it's less. Submaximally, the knockout is less. Um,
When you say maximally,
so here they used the word submaximal when they were just referring to
hmm
like not the maximum contraction as they do with the sensitizing contraction.
hmm
And afterwards, which is probably the important part, you can see that this is increased, so that the contraction force is increased, versus here it's sort of almost stayed the same. So this is like twice, and this is not probably significant. So here I can see sort of what they described here,
what
which
about these?
uh the knockout. So I guess at baseline the knockout is lower. That's why it's lower, is my interpretation.
we could say that it's pretty much the same.
Yeah.
Just because in the passage, right, they said that um within each set of mice, the forces in the baseline twitch contractions were similar.
Okay, I see. Mhm.
So, so like, if it's something like that isn't that much of like this is the difference here is by point one. Um, so, you know, you want to think of that as being pretty much the same. Just like you were able to say like, the point nine and the 1.1 are pretty much the same. But yeah, overall, you see that the major difference is after the sensitizing contraction, which is what, they also tell us in the passage. So, okay, cool. So we see like this proportionality here, in the table. So, AKA, the mutant has a smaller, after thing, because that would be
Yes.
the final twitch contraction. Okay, cool. So any questions here?
Not right now, no.
Okay, awesome. So, okay, so now let's take a look at this question here. So yeah, why don't you, read the question and tell me like, what is it? Yeah, you can read the question.
If skeletal muscle force development precedes the comodulin complex binding MLCK, as described on the passage, which of the following events would occur first after stimulation? I
Okay.
guess I was very confused, and I think I had highlighted this when I was going through it, what they really meant by skeletal muscle force development. Um, yeah, I was confused by just that term in
Oh,
general. Mhm.
so okay, tell me more about like what you're like how you're confused about it.
Yeah, because I was thinking that like the complex binding to MLCK, it's sort of for contraction, sort of like calcium collagen binding MLCK, actin and myosin getting closer together, and boom, you have contraction. And I just didn't really understand what they meant by skeletal muscle force development. Like muscle force development, I always think of it as in the contraction itself, and I don't know what they mean by this, by like contractile strength development, maybe. I don't know. Yeah.
Yeah, you have the right idea. Um, because uh okay, let's say that we didn't have that word. That would be muscle development. That would be very different from if we did have the word, which is talking about the force produced, um, which would be, produced in the contraction. So so you have the right idea there. Um, and also you see, in the passage, they studied the force production, right? All of this, like that's relative force as well. So the forces of the contraction, right? So skeletal muscle force development is the, the force that produces that contraction, right? Okay. And they're saying that this happens before, um, or they're saying if this happens before the CAM complex binds to MLCK. So if that was the case, which of the following would occur, after or first occur first after the stimulation? And the stimulation is like, is this, right? Like, artificially.
Oh, I see.
Hmm.
Oh, okay.
Yep, yep.
Yeah, so when they stimulate, that's when they get they're stimulating, probably depolarizing the neuron. So that's when they're getting the calcium released.
Um, when you say depolarizing the neuron, tell me that again.
Yeah. Um, after the stimulation is like the electrically stimulated.
Mhm.
When you're electrically stimulating, I guess, a neuron here, you would be, I guess, causing probably like a depolarization in the neuron,
Huh?
leading to like calcium being released from the sarcoplasmic reticulum. Is that correct?
Uh-huh.
Right. And
So yeah, go on.
and but this is saying that I'm confused. Yes, I understand this, but I have trouble understanding the question.
Sure. So let's see, right? So we're being told that um that a contraction essentially of a muscle um you know can happen um or rather if it happens before this CAM complex binds to MLCK, then what would happen first after the stimulation? So um so basically uh after the muscle gets stimulated, what can I guess what can happen, I guess, is what they're asking. Or um can you go up on the left? So let me take a close look at this CAM complex thing. So they're saying that the binding of the CAM complex uh to MLCK um causes or precedes the um the actin myosin like interactions, right?
Yeah.
We're saying that so this CAM complex causes, when it binds to, so like when the CAM complex binds to the MLCK, it causes some autoinhibitory region to move away from the active site to be, uh, phosphorylated, um, and then that can cause the actin myosin interactions. So let's think about, what that means. So, um, okay, so we talked about the calcium in the sarcoplasmic reticulum, and that is going to, you know, at the neuromuscular junction, going to cause, release of neurotransmitter like acetylcholine. But if there's, and so that would be like um the thing that happens before the muscle contraction. But if we're talking about like, or rather that's happening at the neuromuscular junction, but if we're talking about just at the level of the muscle, um, then that would be when the actin has the tropomyosin blocking the sites.
Yes.
So is this is pretty tough passage um in that they're just using different words here. So like auto-inhibitory region, I'm pretty sure that's the tropomyosin, right? So here, let me get you. Can you go to this link and yeah, share your screen?
Um, wait, I think need to get to my Zoom. I'll just stop share for a second.
Yes. So this is what happens, at the level of the sarcomere. So we see that on the top left, we see the actin filaments, right? And we see something called, you know, tropomyosin. And yeah, so on the top left, you see that, right?
Yes.
Now, when so like for instance, like that, and that, like are going to be where the myosin can bind. However, it's blocked by the tropomyosin, right?
Yes.
So this is the autoinhibitory thing that they mentioned in the passage. So they're just making it, really like they're the wording that they're using is making it what makes it really confusing. But if you know this, then you know that that's what they mean by the autoinhibitory region. So when calcium is introduced here, the calcium binds to the troponin here, which are these kind of like the beads on the tropomyosin. And when calcium binds to it, like here, then it causes the tropomyosin to rearrange. And as that happens, it exposes the myosin binding sites. Okay.
Yeah.
Now, and now here we can see that, um, this one over here. So we're seeing that we have a myosin filament, we have an actin filament, and the actin, um, you know, has the uh, myosin binding sites, exposed. So what can happen here is that, well, what first happens is that ATP, so it starts off already bound. Oh no, sorry. Um, so it starts off with um actually, yeah, it starts off, let's just say it starts off where it's bound over there. Um, ATP would be something that you would need to unbound this. So, or unbind this. So actually, let me see like what would be a better place to start, because I don't like that being the place that starts it, but okay, let's just start it here then. So okay, so we have, let's just say, yeah, ADP and some phosphate, and it's bound. Um, sorry. Okay, so let's just say that, because okay, basically, I'm just gonna start here. So basically, um, ATP is going to cause the myosin head to not be bound to the actin. Okay. Now, what can happen? So, like, that means that, in the next step here, that ATP can get hydrolyzed when the myosin head is no longer attached to the actin. Now, so now here, the ADP and the phosphate that are bound to that myosin head, right? Um, that happens as the myosin head binds to the actin. So we have it bound to the actin. And then the ADP, and then over here, the ADP and the phosphate release causes the head to change position and the actin to move.
Yeah. Mhm.
Yeah, one sec. I want to get a picture that talks about that's better. So hold on. This is much better. One sec.
Okay. So should I stop share and get the new one?
Yeah, okay, this is better because we can see we can see it like when it's bound and unbound. So okay, so let's just start off, yeah, here at the 1. And this is the myosin cross bridge attaches to the actin myofilament. So you have the myosin head attached to the actin. And yeah, we have ADP and inorganic phosphate. So, okay, when the ADP and the inorganic phosphate is released, it causes this, I guess they're calling it the working stroke, but it's going to like it can also be called the power stroke.
Mhm.
But that what so what's happening here is that this angle is moving like up, so it's causing it to kind of go in this direction, which is going to cause the
Slide.
yeah. So that's the working stroke. The myosin head pivots and bends as it pulls on the actin filament, sliding it towards the M line, the middle line of the sarcomere. So that's where the action of the contraction happens. Now, at this point here, it's still attached to the actin. ATP is needed to detach it from the actin. So so what can help you kind of like help you remember that is when things die, right? Um they exhibit rigor mortis, right? Which is when their muscles are like permanently in a state of contraction. And that's because when they're dead, they're no longer making ATP.
Yeah.
So you actually need the ATP. So like it's like people would think like ATP is what you need to attach it, attach the myosin to the actin, or cause the power stroke. But the ATP is really the thing that detaches the myosin and the actin. So, and think about the rigor mortis, as an example to help you remember that. But yeah, when the ATP attaches to the myosin head, the cross bridge detaches. So it goes back here. And um, and then over here, the ATP can be split into ADP and inorganic phosphate, which causes the cocking of the myosin head. So you have it see. So you have like, so let's say you have like an angle here like that. Let's say that's like a 45-degree angle. Upon ADP phosphorylation, or sorry, hydrolysis, it takes on like a 90-degree angle.
Yeah.
Yeah, the angle is just what I'm using for like uh like the piece of the myosin and the head. So it can be like this, or it can be like that, which means that, compared to, so like for instance, this is the 90, and then this is like the 45.
Yeah.
So, its default position, when it gets detached is at this like 45-degree position. And then as the ATP gets hydrolyzed, it cocks the position. So this is the cocking position.
Oh, I see. Mm-hmm.
That's going to be 90 degrees. And so that's cocking it. So it's 90 degrees. Then it attaches to the actin, right?
Mm-hmm.
And again, you see that it's still 90 degrees here. And then for the power stroke, that is when, so if you imagine like, actin here, when it goes from this to this, right? As this bends down like that, it'll pull that actin over, right?
Oh, yeah, I see that.
So that's why from here, where it's, a 90-degree angle, it changes to, like 45-degree angle, let's say. So it does that. And that's what causes that's the power stroke.
I see. Yeah.
So basically its default state is involves the myosin attached to the actin. Um, and when that happens, the myosin head will produce a power stroke, which is going to cause it to bend, or to bend from 90 degrees to, let's say, 45 degrees. And that, if it's bound already to the actin, it's going to just cause that actin to slide over.
Yeah.
And that's, that after it does that, it's gonna require ATP to then detach from that actin. So like this is the contraction part, and this is like the the relaxation part. And that's, done upon the addition of ATP. And then when the ATP is hydrolyzed, it will return back to the cocked position, which is at a 90-degree angle.
I see.
Hmm?
Yeah.
Yeah. So now, yeah, the passage just is talking about the same stuff. It's just using really, difficult language. Like the tropomyosin, it's calling like the auto-inhibitory site. Um, but yeah, the passage said that, yeah. So any questions here?
I think not right now.
Okay. So yeah, we can go back to that UWorld thing. All right. So yeah, so if we go up on the left for the passage, we can see what we're talking about here, right? So okay. So we're saying that um that when so yeah, calcium is released from the sarcoplasmic reticulum and it binds to this complex that causes a rearrangement of this kinase, in which an autoinhibitory, region moves away, and then that can cause the actin myosin interactions. So, based on what we know about, that all that stuff that we just talked about, like the power stroke and stuff, that's how we can approach this question.
Okay.
So, we want to see, like, so they're saying if the muscle force development um precedes the CAM complex binding to MLCK, what can happen first after the stimulation?
Yeah. Um, well, I think the first thing that happens is this probably.
Tell me, which one?
Right. I think instead of reviewing, I was like, sorry.
What's up?
Yeah.
Oh, wait. Oh, yeah. So no, I wanted us to do it without the
Oh,
answer.
I
But
I I chose it before. That's why I was just checking if it was correct.
got it.
Yeah. And I got to this answer just from like the first step of calcium binding and in order to move away and be able to attach the to the actin filaments,
Huh?
myosin being able to act to the filament. But in that I understand, but I'm still confused by what they mean by muscle force development. Like how can you have like how like what do they mean by muscle force development? This part I'm still confused about.
Got it. So that's the contraction. And then that's specifically the power stroke, because the power stroke is what causes that muscle to contract. And if it's contracting, then it's producing, a force, right? So when they say that, that's what they mean. So when we think about um what can cause a muscle to contract, it's good to be able to like from the first thing to the end, if you can be like so I'll just tell you from the beginning to end. So the first thing that happens, all right, is the action potential from the motor neuron, like is going to travel on through the neuromuscular junction into the muscle, right? And that happens because the calcium that's released from the sarcoplasmic reticulum gets taken up by the motor neuron, and the motor neuron then releases acetylcholine uh, neurotransmitter, which gets attaches to the motor end plate of the muscle, which then opens up, you know, the ligand gated sodium channel, so sodium can influx and then cause the muscle to undergo depolarization and take on that action potential. And and then that means that and then calcium, like, so what I just described is like the neuron to the muscle, but now from the muscle to the muscle to contract, you're going to have, it's going to start off with calcium again, with calcium binding to the troponin. And when it binds to the troponin, it'll cause a conformational change that will move the tropomyosin away from the actin's myosin binding sites, allowing the myosin to bind to the actin, forming the cross bridge. And the myosin head pivots, which is when it goes from the 90 degree to the 40.
Mhm.
And that is the power stroke, which causes the actin filaments to move towards the center of the sarcomere, right? And that's the contraction. That's the muscle force development.
Oh, I see.
Yeah. And then after that, ATP comes in, binds to the myosin head, causes it to release the actin. And then the ATP, but it's still in a 45-degree angle, right?
I see.
So, so, um, so once the ATP gets hydrolyzed, it can return to its 90-degree angle.
I have a question
Yep.
about disorders.
Yep.
So, I'm remembering that in the passage they're stimulating it electrically. So, is this why in this question they're preceding the order? So they're changing it as if you were to contract before you had that stimulation. So before the neuron level, so is that why? Like, is it normally like you have this contraction and then you have the depolarization? And and but because in this case they experimentally are like inducing the contraction, that you don't have it, but normally it would be like it would normally precede this.
Yeah. So let's say that so the stimulation part means that we've already, because it's really like through neurons, right? And then neuron at the neuromuscular junction to the muscle, and then the muscle to the muscle to contract it, right? So, if they did not say this, right,
Mhm.
then what would be a good answer?
Um,
So let's think about what I kind of wrote here. So neuron to neuron is just, passing of an action potential through voltage gated channels, right? And
okay.
then neuron to muscle, right? I'll leave that for now. But muscle to muscle will, well, okay, sorry. I'll go over the neuron to muscle. Neuron to muscle, that's when you have the sarcoplasmic reticulum releasing the calcium, right?
Yeah.
And then that does, that allows myosin and actin to bind. And then we're just on the level of the muscle. And this is when the calcium can bind to the troponin to cause that cross bridge,
Yeah.
right? So if this was not mentioned, right, what would occur first? Well, first is an action potential traveling from a neuron to another neuron, but we don't see anything like that here. So next would be what happens at the neuromuscular junction. So this would be our answer if this wasn't saying that what would occur first after stimulation.
But
Mhm.
I see. But in C, like I guess I like
Oh
when it's
crap! It
the
says
update,
reuptake.
I
Yeah, I
always
didn't.
think of
I didn't see that part. What that part? So sorry. Like, yeah, if that wasn't there. So just what I'm trying to say is that if it did not say after stimulation, then what would happen is sarcoplasmic reticulum uh will release calcium. Yeah, I didn't see the reuptake part, but I just want you to see that that's the first thing that happens because it happens at the neuromuscular junction. Now, if they said stimulation has already occurred, this muscle is already stimulated. The neuromuscular junction part's already happened. So now we're only on the level of what happens to the muscle itself.
I see.
So that is going to be the binding of calcium to the troponin because that's going to form the cross bridge.
Yes,
Mhm.
I see.
Yep.
Mhm.
Yeah. So, uh, you know, so yeah, so the sarcoplasmic reticulum reuptake of the calcium, that's going to be like way, way at the end, right? So that's out. Hydrolysis of ATP by myosin. What can you tell me about that?
The hydrolysis of ATP is a power stroke usually. So that's the contraction itself.
Oh, so the hydrolysis of the ATP, that's going to be the one that turns it, that cocks it.
Yeah. Is that's the power stroke, right? Or
Oh, sorry, that's the cock. So like so basically, let's start off with it being like at this angle.
yeah.
And this is the actin and it's not bound, right? Now, yeah, ATP is here. But now when ATP gets hydrolyzed, it causes the myosin head to cock, which means go to be at a 90-degree position, right?
Yes.
So
Mhm.
now, so this is the cocking step.
I see it's
Then
separated.
it's going to be attaching step, like forming the cross bridge to attach.
Yeah.
Then it's going to be still attached, but it's going to go from the 90-degree to the 45-degree, and that's the power stroke.
I see. I see.
And then after that, um, through the addition of ATP, it'll be detached.
I see.
So you can think of it like in four kind of steps. Like the first step is, well, what I like, the different pictures choose different things to be the first step, but I think it's easiest to think of the first step as um as the myosin and actin not being connected, and the myosin is at a 45-degree angle. Um, and when and this is with ATP. So when the ATP gets hydrolyzed, so we just want to think about two things. You want to think about attaching to actin, detaching from actin. And then the other thing we want to think about is 90-degree angle, which is the cocked angle, and or 45-degree angle, right? So if we start off with 45-degree angle, not attached, right, to the actin, then what can happen first is that it would have to go to the 90-degree position. So it'll have to cock first. Then what can happen? Well, it's not going to go back to 45. So what can happen next is it'll attach to the actin. So that's when it attaches. And then after that, it can go back to the 45 position, and that's the power stroke. Okay.
I see.
Yeah. So just remember that attaching and detaching, um, well, in order to attach, you want the myosin, you need the myosin head to be cocked, right? And um, for the power stroke, you want the myosin head to be both connected, but no longer cocked. So it goes back down to the 45-degree angle.
I see.
And then you want it to detach. So you're either gonna have you're gonna have like a step where you cock it, and then after you cock it, you attach it, and then after you attach it, you uncock it, which just means it goes back to 45 degrees. And then when you're done uncocking it, you can detach it. And then before you attach it again, you have to cock it again. Yep.
Yeah.
Yeah. So, okay. So for instance, D, hydrolysis of ATP by myosin, that would cause what to happen out of these four things?
Um, it's to get on probably this one.
Where?
Are you able to see my
Yep.
mouse? So let me think through it first.
Yeah, yeah. And also like, if the diagram is confusing, you can tell me.
Yeah. So hydrolysis of ATP. So ATP, you said, is you need to have it around in order to relax.
Wouldn't you
And
say
so
relax?
relax,
Yeah, what do you mean?
like stop the contraction.
Well, um, so you need the ATP to cause the myosin to detach from the actin.
Yeah. Yeah. Um, yeah. So I was thinking in terms of when you have when if like you need like I'm imagining like the actin being attached to the myosin as contracting, but is that incorrect? Is it the actual this position, the 45-degree angle, that's contraction?
So
Okay.
the contraction is, let's see if I can like demonstrate perhaps. So let's see. Okay. Let I guess I'm just gonna use like my hand. So uh this hand is the actin,
Mhm.
and then this will be the myosin head.
Mhm.
So let's start off with um them not attached
Okay.
and the myosin head being like at a 45-degree angle. So that's when you have ATP.
Yeah.
Now,
Ah, I see. So ATP hydrolysis is gonna take it to that cocked position.
yes,
And then uh I guess calcium troponin moving is gonna allow it to attach.
yep,
And then I think it's like a phosphate release, though. Maybe. Um, I have in my mind something of like a inorganic phosphate being like released that's moving and actually causing like like the power stroke. Is that correct?
So, I don't know about the inorganic phosphate leaving part, but you're right in that, yes, when it attaches, right? Um, it will like what the ATP hydrolyzes into, like the ADP and the inorganic phosphate, like when that stuff is, you're done using it, it's going to go like it'll uncock itself, so it'll go from the 90-degree angle to the 45-degree angle, but since it's attached, it'll pull. You see how my left, like my arm, this arm
Yeah.
is going from this way to this way.
Yeah,
So that's going to be the actual power stroke part.
I see. So
Yep.
this was helpful. Hands are
Yeah.
helpful.
Yeah, exactly, exactly. And then yeah, just being able to see it in terms of like like pretty much just there's only two things you could do. You can either attach to the actin or detach from the actin, and the other two things that you can do is you can either be at the cocked position or at the uncocked position.
Okay.
So yeah, so you just want to like alternate between those. So you want to start like I think it's best to start off um at the uncocked position, unattached. Okay. And then that's when you have ATP. When ATP hydrolysis happens, right? You still have it detached, but you cause the myosin head to cock. And then when it's cocked, it can attach. And when it's attached, it can be uncocked, which will cause the power stroke. And then it can be detached.
Okay. I see. I think it,
So cocked position will happen before it attaches. And
yeah.
uncocked will happen um, before it detaches.
Yeah.
Okay.
Yeah.
Yep. Awesome. So yeah, so this dehydrolysis of ATP by myosin, that's going to be like basically from here to here. It'll cause the cocking.
Yes. I see.
Okay. Yep. Yep. Does that make sense?
It does.
Let me see what they wrote down here.
Yeah.
Yeah. Okay, there we go. So, okay, let's see. Um, there's a lot of extra stuff, but okay, over here we see this is the starting position where it's 45 degrees, not attached. ATP gets hydrolyzed, causes it to be in a 90-degree position, and um, wait, ATP hydrolysis causes that and that. Yeah, I think this is just making it more confusing, but pretty much exactly what we just talked about.
Yeah.
Yeah.
It's good.
Hmm.
Oh wow. Yeah.
Oh, do you have more questions for this passage?
For this passage, I think what was it? 11 through 16, and that was the first one. I don't, yeah, I don't remember. This was like Monday, or I don't remember exactly, but calcium most likely initiates potentiation by directly. Mm. It's hard to see because I have my previous answer. I wish I could take that off. When wanted to promote movement of tryptomyosin on the development. So we talked about how calcium is like is allowing the myosin and actin to interact um by like moving away the troponin, which is like blocking usually that interaction of actin and myosin. Um, calcium binds troponin. So not this, not B. And I think we talked about how so it's either A or C for sure, I think.
Okay, so tell me what happens. So I drew this thing again.
Yeah.
Um, I just want to make sure that it makes sense what I'm drawing here. Tell me what I'm trying to like kind of say with this.
I think you said neuron to neuron, then neuron to muscle, and then muscle to muscle.
Mhm,
So like from the neuron depolarizing
mhm.
and calcium um entering the muscle, and then the muscle calcium binding in the muscle and causing the contraction in the like muscle to muscle.
So yeah, so this is when you have the calcium binding to the troponin.
Yes.
This is when you have the calcium doing what?
Allowing for the like actin myosin interaction, so allowing for the contraction to take place.
So that would be like here, right?
Yeah.
With the muscle.
Yeah.
So at the neuromuscular junction, that's when the calcium, gets released from the sarcoplasmic reticulum and then gets taken up by the neuron.
Yeah.
And the neuron can release its neurotransmitter like acetylcholine.
Mhm.
So for this passage, we want to be aware of calcium playing a role in both of these things. Because for this question, right? These choices refer to the muscle to muscle thing, meaning when calcium is used uh, to bind to troponin, causing the tropomyosin to move, allowing the myosin to heads to bind to the actin.
Yeah.
Whereas this stuff is what's happening at the neuromuscular junction, and that's more along the lines of calcium, you know, uh, what is it? Causing the um action potential to reach the muscle.
I see.
So yeah. So so okay, they're saying based on the passage, calcium most likely initiates potentiation by directly doing what? So let's think about yeah.
I think I would, can you see what I'm like?
Yep.
I think it's probably referring to this part of like causing the I would keep my answer actually. Binding calmodulin to promote intramolecular movement of MLCK. Because it's sort of binding it and causing it to change conformation to not be inhibited.
Okay. So since we're talking about potentiation, right? And they said that potentiation is when you have a larger skeletal muscle force production. Um, despite having the same depolarizing stimulus, right? We know that this is going to be something that, is happening at the neuromuscular junction rather than within the muscle itself. And so that means we can get rid of A and B. Now, C and D, tell me why you prefer C.
So promote and prevent. So it's I prefer C because it is causing it to do something instead of like uh uh yeah, I would just stick with C. Just promote versus prevent because it's binding and it's causing it to be like activated.
Yeah. So we see that MLCK is regulated by CAM, the binding of CAM, which occurs when CAM binds to the calcium released from the sarcoplasmic reticulum. So we are talking about that aspect of it. Afterwards would be, this stuff, right? But yeah, we're talking about potentiation, which is going to be, yeah, like prior to this. So this is like more related to you know, the calcium being released from the sarcoplasmic reticulum at the neuromuscular junction. So yep, I believe that will be your answer, yeah.
I guess we can check.
Sure.
Yeah.
Yep.
I see.
Mm-hmm.
I guess should we go to another question?
Yep. All right.
I remember reviewing this and I think I chose the wrong one. That's one. And it was because I was confused between fast twitch and slow twitch. And if I remember correctly, fast twitch, slow twitch. Fast twitch means fast contraction, right?
Okay.
I think. Um, which means rapid decay.
Oh, 1 second. I'm just gonna use the bathroom real quick.
Yeah. Actually, I'll also be back. So show me. I can find Zoom.
All right. Cool. Okay. Yeah.
Yeah.
Walk me through your thoughts here.
I haven't read it. Sorry, I also used the restroom.
Oh, gotcha. Okay.
Just grayed out yeah. So we were talking about fast twitch and slow twitch and like the differences between them. And so I was trying to fast twitch. I don't know if this is correct, but I guess we'll find out after I'm done trying to see. So like for me, fast twitch would be like fast contraction, which would mean that like higher like contractile strength, but they would decay faster as well compared to slow twitch. Is that correct?
When you say decay, tell me what you mean.
Yeah, I'm thinking sort of as in terms of like like aerobic versus anaerobic and like long term versus short term use.
Okay.
Um but I guess maybe I'm precipitating myself. Maybe I should look at the
Oh no, no. So I tell me more about what you just said about anaerobic and aerobic in reference to fast twitch and slow twitch.
yeah. Um so um slow twitch would have like less contractile strength, maybe. But they would be able to be used long term. So I normally associate them, if I'm thinking correctly, with aerobic
Yep.
exercise
Yep.
versus anaerobic. I would go with like the fast twitch, maybe.
Yeah.
And like a lot of contractile strength, but the decay is a lot quicker, like easily fatigue. Um, yeah.
I see what you're saying by decay. Yeah, that's perfect. So yeah, slow twitch muscles can last a lot longer because they are utilizing oxygen, so that's the aerobic part. But of course it's slower and you know, don't generate as much power. Um, yeah, and then the fast twitch is known to generate a good amount of force very quickly or power. Um, but yeah, it would have like larger refractory, type of delay afterwards, right? So okay, so now let's see. So they're asking us, so what are they asking us here? Like, yeah, so what are they asking us?
So they're asking us to think of why they chose um it to be in fast twitch muscles instead of slow twitch muscles.
Yeah, okay. So yeah, let's go through these choices.
A says higher rates of phosphorylation and dephosphorylation. I don't know. I normally don't think of phosphorylation or dephosphorylation in this between fast twitch and slow twitch. So I guess I would move on to B and read it as well.
Oh,
Uh
so well, we mentioned in the passage stuff about, well, I mean, what's the MLCK? What is that again?
it's a kinase. Uh
So kinase must have to do with phosphates, right?
yeah, I guess I just mentioned, so I just discarded it because like it's usually not a distinguishing factor between fast twitch and slow twitch.
I
Uh
see, I see. So
unless
go on.
Yeah. Unless I'm wrong, but I normally don't think that's one of the like differentiators in fast twitch and slow twitch.
Well, let's see, right? So they're talking about potentiation of skeletal muscle force production by MLCK in fast twitch muscles is so like yeah, so like this is the so like yeah, well this is the question, like this is just you know the first part of a sentence that we're supposed to complete with the answer choice.
Okay.
But what characteristic of MLCK activation? Well, we know that what it we know that what it does is phosphorylate something like phosphorylates RLC. Um and then they said that that MLCK can be activated, um, by the CAM kind of complex. So okay. So let's I guess yeah. So let's check out B. Let's move on to B then.
Aided by lower comodulin concentrations than in slow twitch muscles. I feel like if it were aided by lower, then if they're describing that this is part of what's causing the potentiation, the CAM,
Okay.
maybe they wouldn't want the, I don't know, aided by, I guess my thought process is maybe they would want it to be higher in the ones that they're observing because this is involved in the in the activation of the MLCK. Unless it's beneficiary to have it be lower, but I feel like I'll keep reading C and see what happens. Um, Easier to detect than in more quickly fatigued slow twitch muscles. I think that would be a valid reason, maybe.
Okay.
But unless it's quickly fatigued slow twitch are not quickly fatigued, right?
Mhm.
They are
Yeah.
they are long lasting. So this is incorrect, although it would be a valid reason. Facilitated by the presence of higher myoglobin levels. So it's factually incorrect as well, right? Because I think slow twitch, which are involved in aerobic, which have a lot of mitochondria, probably also have higher myoglobin levels. So I think it's also factually incorrect.
Uh-huh. So, good. You should have, yeah, myoglobin is yeah, used for oxygen storage. And that should be higher in slow twitch. Good. So yeah, we're already at 50-50.
Yeah. So A or B.
Mhm.
Potentiation is dependent. I guess if it's more phosphorylated, then it would be more activated, right? So I would go for A between A and B just because of that. Because normally if you have more dephosphorylation, you would be more inhibited if you want to study the MLCK and kinase.
Yeah. So so for B, I want you to tell me what makes B not a good choice.
Agent. Yeah. If you have less of calmodulin, which is this complex with calcium that activates MLCK, then you would your enzyme that you're studying will be in its inactive form. So it's sort of the same as like the more dephosphorylated. So they're actually in opposition A and B. So because it would be in a more inactive form, it would be you wouldn't be able to study potentiation as well.
Yeah,
That's
because it's the binding of the calcium to the calmodulin that um that activates MLCK, right?
yes.
And B is just talking about lower like calmodulin concentrations. And yeah, that shouldn't really, I mean, we don't see anything in the passage that talks about how you know, like fast twitch and slow twitch muscles have different CAM concentrations. So, so yeah, it's not the CAM concentration that really matters. It's the binding of the calcium to the CAM, which causes the MLCK to be activated. So yeah, so that would be out. And yeah, dependent on higher rates of phosphorylation, um, uh, and lower rates of dephosphorylation than are present in slow twitch muscles. Um, so we know that the phosphorylation, so we know that MLCK uh phosphorylates the RLC, and the RLC phosphorylation, like when that gets phosphorylated, that will facilitate actin binding to myosin, right? And that leads, and the actin binding to myosin forms a cross bridge, which can then produce the power stroke, right? So, and the power stroke is what determines the muscle force production, right?
Yeah.
So, yep, it will be A.
Yeah. It's like the one I completely like crossed out at the beginning. Yeah.
Mm-hmm. Yeah. So I'm going to have to leave pretty soon,
Yeah.
but let's see if
Yeah, that's good. It's actually been a long session.
oh yeah, I mean I've had much longer, but let me just take a look at 15 and 16
Okay. Sounds good.
because maybe it would be something I could go over quickly. Um, what do you think of this one?
My force production was not increased above baseline until after the sun. So it's asking me this. So why the force production, relative force, was not above baseline until after the sensitizing contraction was completed. So why it wasn't in, let's see. But they were so it's just asking me why they needed the sensitizing contraction in order to increase the force. Is that what they're asking me?
Mm-hmm.
Rate of ATP synthesis. So not this.
Mm-hmm.
Phosphorylation. I don't see why ATP synthesis would be needed for this. Hydrolysis of ATP is a slow process. It's C. I would keep C just because it is the phosphorylation state that is changing
Mhm.
with this that is leading to the potentiation.
Mhm.
Yeah. Yeah. Yeah, that took some time.
Mhm. No, it didn't take too long. Yeah, and then let's just look at 16. Oh, I guess this is like a math
Yeah, that's an easy one.
problem. Okay, so you got this one.
Yeah.
Okay, awesome. So let's see. So okay, so we talked about, I mean, of course you could just look at the recording, but yeah, we talked about buzzwords, so words that you see in a passage that uh that are likely going to be asked about, or just things that you have to know. So like today we saw kinase. You want to know that kinase is, you want to know the enzyme classes, so it's a transferase, and it uses STY, um, amino acids that have hydroxyl groups in them. And whenever you see a hydroxyl group, I want you to be thinking nucleophile. Carbonyl group, I want you to be thinking, electrophile. Um, and then I want you to see how a phosphate group uh, acts like an electrophile, because it's somewhat similar to like the carbonyl, the double bonded oxygen and stuff. Um, and so that's how kinases can use STY to, transfer phosphate groups. Oh yeah, and then so yeah, you understand the sequence. Like, yeah, I would like know the entire sequence of um like for instance, like action potential from motor neuron to the neuromuscular junction. Calcium can be released from the sarcoplasmic reticulum and through the T tubules and get taken up by the motor neuron, which will cause the motor neuron to release acetylcholine, which can then go into the neuromuscular junction and bind to ligand-gated channels at the motor end plate, which causes the sodium influx to cause that to depolarize. And that's at the neuromuscular level. And then at the muscular level, so like the way, that I separated the two, like neuron to neuron, neuron to muscle, muscle to muscle. Um, the muscle to muscle stuff, that's where we talk about the sarcomere. And then yes, for that, you want to know that, um, you know, it depends on where you want to start, but I like to start, I think it's easier to start at um 45-degree position for the myosin head, and it's not attached to actin. And this is with ATP. ATP gets hydrolyzed. So what happens first? Does it attach to actin first, or does it attach, or does it go to the cocked position? Um, Cocked position, right? And then it will attach, right? And then it won't detach, right? It'll go back to the 45-degree position. So it will, and when it does that, it brings the actin down, like across, causing that contraction. And then this is called the power stroke or the work stroke. This is where the force, is generated. Um, so after it's done that, you want to detach it from the actin, and you need ATP for that. That's why if you are dead and you don't have ATP, then your muscles are in that perpetual state of contraction. So yeah, that's pretty much most of what we went over today. So yeah, I would say to, like, yeah, I guess like just review this stuff, and just to know it and to see it, be able to understand it in the different ways. And um yeah, any other things?
No, but I think last time you said you were going to send me like a schedule.
Oh yes, yes, yes. Yeah. Yeah. Yeah, okay. So yeah, let me write that. So okay, I'm gonna try to get that to you by the end of like this weekend. Um, so yeah, it'll be like I'll right away share like the calendar thing with you. Um, so like it'll be like your schedule in the calendar format. And I'll add more things to it after, but yeah, I'll share that calendar with you. And oh, and then I share the drive with you. Um, and yeah, I think that's. Oh yeah, we didn't get to go over genetics, but I guess we can like do that next time. But yeah, I think you're doing a good job, and yeah, just continue with, finishing up UWorld and yeah, having questions for, when we meet next. By the way, yeah, when do you want to meet next?
Um, next next weekend would be good too. Like next
Uh-huh.
Saturday
All right, let's see. Um,
or Sunday if that works better.
no, I can do Saturday. I can even do like earlier, like um you were in the West Coast, Central?
East Coast.
Okay, okay. Um yeah, okay. Do you want to do Saturday the 24th at 3 p.m. Eastern?
Yeah, 3 p.m. works well.
All right, perfect. And yeah, so just continue with what you're doing in regards to UWorld and have like stuff that you want to go over when we meet. Um and yeah, I think you're doing a good job. Okay.
I think for me the passage reading and walkthrough was very, very good. I think it was nice.
Yeah, we can do that because like yeah, I can show you more stuff uh in terms of like how to like yeah, analyze really long dense passages like that, like with the proportionality thing and the schematic thing and yeah, I can show you that stuff, as we continue to work together. So yeah, awesome. Okay. So yeah, anything else?
Nope.
All
That's it.
right, awesome. So I will see you next week. Good luck. Email me if anything. And yeah, have a good weekend. Okay.
Thank you.
All right, take care. Bye.